Hello everyone! In this lecture, we are going to discuss RNA transcription. The transcription is the first of several steps of the DNA-based gene expressions. During transcription, a DNA sequence is read by an RNA polymerase, which produces a complementary anti-parallel RNA strand called a primary transcript. Transcription proceeds in the following general steps. RNA polymerase, together with one or more general transcription factors, bind to the promoter region of the DNA. RNA polymerase creates a transcription bubble which separates the two strands of the DNA helix. And the RNA polymerase adds RNA nucleotide, which are complementary to the nucleotides of one DNA strand. RNA may be further proceed, including polyadenylation, capping, and splicing. The RNA may remain in the nucleus and exist to the cytoplasm through the nuclear pore complex. RNA polymerase is the enzyme that is responsible for copying a DNA sequence into an RNA sequence. During this process, we call it transcription. There are three RNA polymerases. RNA polymerase 1 is in higher eukaryotes the RNA polymerase that only transcribes ribosome RNA. RNA polymerase 2 is a multiprotein complex. It is one of three RNA polymerase enzymes found in the nucleus of the eukaryotic cells. It catalyzes the transcription of the DNA to synthesize precursors of mRNA and most snRNA and microRNAs. In eukaryotic cells, RNA polymerase 3, also called polymer 3, transcribes DNA to synthesize ribosome 5S rRNA, tRNA, and other small RNAs. Transcription involves four steps. Initiation. The DNA molecule unwinds and separates to form a small open complex. Elongations. DNA polymerase moves along to the template strand, synthesize mRNA molecules. Termination. RNA polymerase will keep transcribing until it gets signals to stop. The process of ending transcription is called termination, and it happens once the polymerase transcribes a sequence of DNA known as a terminator. Processing. All RNAs are originally transcribed from DNA by RNA polymerase, which are specialized enzyme complexes. But most RNAs must be further modified or processed before they can carry out the rules. Thus, RNA processing refers to any modifications made to RNA between its transcription and its final function in the cell. Prokaryotes utilize one RNA polymerase for all transcriptions of types RNA. In contrast, eukaryotes utilize three slightly different RNA polymerases. In prokaryotes, which lack membrane-bound nucleus and other organelles, transcription occurs in the cytoplasm of the cells. Bacterial messaging RNA is generally polycistronic. Polycistronic describes a type of messaging RNA that can encode more than one polypeptide, which is separated within the same RNA molecules. Each of the three RNA polymerases in eukaryotes is responsible for transcribing a unique type of RNA. A mRNA molecule is said to be monocistronic in eukaryotic organisms, which means mRNA contains the genetic information to translate only in one protein chain. Messaging RNA is a large family of RNA molecules that convert genetic information from the DNA. The RNA polymerase enzyme transcribes the gene into primary transcript mRNA, which is known as pre-mRNA. Transcription start site usually have a TATA box, but not always. The TBP, which called TATA binding proteins, binds changing DNA structure, recruits transcription factors to proteins, and then RNA polymerase. Here is a summary of the transcription process. So initiation, the double helix of the DNA is unwound by RNA polymerase, which docks on the end of the DNA at the special sequence of base. In RNA polymerase II dependent transcription, there are 6 general transcription factors: TF2A, TF2B, TF2D, TF2E, TF2F, and TF2H. The TF2D is the first component to bind the DNA due to binding to the DBP, while TF2H is the last component to be recruited. After recruitment of the DNA polymerase, the transcription starts. RNA polymerase transcribe the DNA information to DNA until recognize stop signal poly-A sequence. During the elongation process, the RNA polymerase moves downstream unwound to the DNA. As the double helix unwinds, ribonucleotide base A, C, G, and U attach themselves to the DNA template strand, the strand being copied by complementary base pairing. RNA polymerase catalyzes the formation of the covalent band between the nucleotide. In the wake of transcription, DNA strand recoil into the double helix. RNA splicing in molecular biology is a form of RNA processing in which a newly made precursor messaging RNA transcripts is transformed into mature messaging RNA called mRNA. During splicing process, the intron, which called non-coding region, are removed by exons are joined together. Splicing is carried out in a series of reactions which are catalyzed by the splicensome, a complex of small nuclear ribonucleic proteins. The major splicesome splice the introns containing GU at the 5' splice site and AG at the 3' splice site. It is composed of the U1, U2, U4, and U6 slow RNAs, and it is active in the nucleus. In most cases, the splicing removes intron as a single unit from precursor mRNA transcript. In briefly, step 1, the U1 binds to the 5' splice site and the U2 binds to the branch site. Step 2 is the splicesome was assembled and start cleaved the transcript at the 5' splice site. The 5' end of the intron is joined to the A in the branch site to form a letter structure, and the U1 and the U4 are released. The step 4 is then the 3' splice site is cleaved and the 5' end of the intron 2 is joined to the 3' end of the exon 1. The Laria-shaped intron is released along with the slow RNAs U2, U5, and U6. The 5' caps is a special altered nucleotide and the 5' end of the same primary transcripts, such as precursor messaging RNAs. This process known as mRNA capping. The capping process. One of the terminal phosphate group is removed by RNA triple phosphatase. leaving a biphosphate group. The GTP is added to the terminal to biphosphatase by mRNA guanine transferase, losing a pure phosphate from the GTP substrate in the process. The 7-nitrogen of the guanine is methylated by mRNA guanine N7 methyltransferase, resulting the cap. The cap adjacent modification can occur normally to the first and second nucleotides, producing up to the real cap 2, cap 1, and cap 2 of the messaging RNA. Polyadenylation is the addition of the poly-A-tail to the messaging RNA. The poly-A-tails consist of multiple adenosine monophosphatase. Polyadenylation is part of the process that produces mature messaging RNAs from the translation, and is therefore forms part of the larger process of gene expression. The process of polyadenylation begins as the transcription of the gene terminates. The 3' most segment of the newly made mRNA is first cleaved off by the set of proteins. These proteins then synthesize the poly-A-tails and at the end of mRNA. The poly-A-tail is important for the nuclear transportations and stability of mRNA. The transportation of messaging RNA from the nucleus to the cytoplasm is an essential step of the eukaryotic gene expression. The mature mRNA are recognized by their processed modification and then exported through the nuclear pore by binding to the cap binding proteins CBP20 and CBP80, and then exported the complex formed. Here is some reference, and thank you for your attention.